Hello everyone, I am Dr. Sirajul Faisi. I welcome you in my YouTube channel. Today in this video, I am going to talk about enzymes. You know friends, enzymes play an important role in chemistry. So I will start our lecture from introductions, little bit chemistry, then classifications, mechanism of enzyme action, enzyme kinetics, inhibitions, activation, and specificity. So, chemistry behind it. Enzymes are biological catalysts that speed up the rate of the biochemical reactions. My friend, most enzymes are three-dimensional: globular, protein, tertiary, and quaternary structure. And some special RNA species also act as enzymes and are called ribozymes. Example, hammerhead ribosome. Now, friends, structure of enzymes. You know, friend, the active site of an enzyme is the region that binds substrates, cofactors, and prosthetic groups and contains residue that help to hold the substrates. Active site generally occupy less than 5% of total surface area of enzymes. Now, active site has a specific shape due to tertiary structure of protein. A change in the shape of protein affects the shape of active site and function of the enzyme. So active site can be further divided into active site, binding site, and catalytic site. It chooses the substrates and bind it to active site. It performs the catalytic action of enzymes. Now friend, cofactors. Cofactor is the non-protein molecules which carries out chemical reaction that cannot be performed by standard 20 amino acids. So cofactors are of two types: organic cofactor and inorganic cofactor. Cofactor in active protein, without the cofactor attached, the protein is not active. Now you can see L1 and L2 binding site, active protein, cofactor binding activate the protein. So inorganic cofactors, these are inorganic molecules required for the proper activity of enzymes. For example, enzyme carbonic anhydrase requires zinc 2+ for its activity. Hexokinase has cofactor magnesium 2+. Organic cofactor, these are the organic molecules required for the proper activity of enzyme. For example, glycogen phosphorylase requires the small organic molecule pyridoxal phosphate. So types of organic cofactor: prosthetic group and coenzyme. What is the prosthetic group? A prosthetic group is a tightly bound organic organic cofactor. Example, flavin, heme groups, and biotin. Coenzyme, a coenzyme is loosely bound organic cofactor. Example, NAD+. So, friend, an enzyme with cofactor removed is designated as apoenzymes. This is new term, apoenzyme. The complete complex of protein with all necessary small organic molecules, metal ions, and other component is termed as holoenzymes of haloproteins. So my friends, what is the meaning of substrates? So the reactant in biochemical reaction is termed as substrate. When a substrate binds to an enzyme, it forms an enzyme-substrate complex. So site of enzyme synthesis. Enzymes are synthesized by ribosomes, which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzyme is carried by DNA. Amino acids are bonded together to form a specific enzyme according to DNA's codes. So endoplasmic reticulum and ribosome, you can see in this picture. Intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained in the cell for use of cell itself. They are found in the cytoplasm, nucleus, mitochondria, and chloroplast. Example: oxidoreductase catalyzes biological oxidations, enzyme involved in reductions in the mitochondria. Extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. So example, digestive enzymes produced by the pancreas are not used by the cell in the pancreas but are transported to the duodenum. So now friends, what is the characteristics? Enzyme speed up the reaction by loading the activation energy of the reaction. Their presence does not affect the nature and properties of end product. They are highly specific in their action, that is, each enzyme can catalyze one kind of substance. A small amount of enzyme can accelerate chemical reactions. Enzymes are sensitive to change in pH, temperature, and substrate concentration. Turnover number is defined as the number of substrate molecules transformed per minute by one enzyme. So, catalyzed turnover number 6 into this 106 per minute. So my friends, nomenclature of enzyme. An enzyme is named according to the name of substrate it catalyzes. Some enzymes were named before a systematic way of naming enzyme was formed: pepsine, trypsin, and renine. By adding suffix ASE at the end of the name of the substrate, enzymes are named. Enzyme for catalyzing the hydrolysis is termed as hydrolase. For example, maltose water, maltase enzyme, give the glucose and glucose. So enzyme substrate products: lactose, lactase, glucose, galactose, maltose, maltase, glucose, cellulose, cellulase, glucose, lipids, lipase, glycerol, fatty acid, starch, amylase, maltose, protein, protease, peptides, and polypeptides. So my friends, I hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.